We humans share a very close relationship with flowers. They have absolutely enhanced our daily lives. But to a plant, a flower is an organ of sexual reproduction. The androecium, consisting of the stamens, is the male reproductive structure in the flower, and the gynoecium, consisting of the carpels, is the female reproductive part. Let's understand in brief the process of sexual reproduction in the most diverse group of land plants, the angiosperms. The stamens, which are the male reproductive parts of the flower, each consist of a long stalk called the filament. At the top of the filament is the fertile portion of the stamen, which is the anther. Each anther is a bilobed structure consisting of 4 microsporangia or pollen chambers. The pollen chambers consist of the sporogenous tissue, which divides to form the microspore mother cell. As the anther develops, the microspore mother cell divides meiotically to form the microspore tetrad. The microspores separate out to form mature pollen grains, which represent the male gametophyte. A mature pollen grain consists of 2 cells, the vegetative cell and the generative cell. If you touch the anthers of flowers such as the hibiscus or the tulip, you will find a yellow powdery substance sticking to your hand. These are the pollen grains or the male gametophyte of the flower. Now, let's look at the formation of the female gametophyte inside the carpel. The swollen basal part of the carpel is the ovary. The ovary contains a hollow chamber called the locule. The locule consists of many ovules, also known as the megasporangia. Inside the ovule is the megaspore mother cell, which undergoes meiosis to form 4 haploid megaspores. Out of the 4 haploid megaspores, 3 degenerate and only 1 remains functional. This is called the functional megaspore. The functional megaspore divides mitotically to form the 2 nucleate, the 4 nucleate, and the 8 nucleate embryo sac, which is called the female gametophyte. The female gametophyte consists of 2 synergids, 1 egg cell, 3 antipodal cells, and 2 polar nuclei located in the large central cell. The mature female gametophyte is thus said to be 8 nucleate but 7-celled. The male and the female gametes are produced in separate structures, the pollen grain and the embryo sac respectively. How are these 2 non-motile gametes brought together? This is achieved by pollination, which is the process of transfer of the pollen grains from the anther to the stigma. Abiotic agents such as wind and water bring about pollination. Pollination by wind is known as anemophily, and pollination by water is known as hydrophily. Biotic factors that help pollination are insects such as honeybees, butterflies, beetles, and wasps. Pollination by insects is known as insect pollination or entomophily. Pollination by animals other than insects such as birds, squirrels, bats, and snails is known as zoophily. Pollination is of 2 types, self-pollination and cross-pollination. Self-pollination is the transfer of pollen from the anther to the stigma of the same flower, or between flowers of the same plant. Cross-pollination, on the other hand, is the transfer of pollen grains from the anther to the stigma of a different plant belonging to the same species. After the pollen grain reaches the stigma, it germinates to form the pollen tube. Inside the pollen tube, the generative nucleus divides mitotically into two male gametes. The vegetative nucleus, also known as the tube nucleus when inside the pollen tube, degenerates after reaching the ovule. On reaching the embryo sac, the pollen tube disintegrates and the two male gametes enter the embryo sac through the synergids. This causes the synergid cell to rupture. One of the two male gametes fuses with the egg cell to form the diploid zygote. This process is called syngamy. The second male gamete fuses with the two polar nuclei to form a triploid nucleus or the primary endosperm nucleus. The formation of the primary endosperm nucleus involves the fusion of three haploid cells and is thus called triple fusion. Since there are two fusions, syngamy and triple fusion, taking place inside the embryo sac, the fertilization is known as double fertilization. The double fertilization event is seen only in angiosperms. After the process of double fertilization, the primary endosperm nucleus divides repeatedly to form a triploid endosperm tissue, and the zygote gives rise to the embryo. The endosperm is rich in starch and other food reserves. The reserved food material in the endosperm is used for the development of the embryo. After fertilization is accomplished, the ovule increases in size. The integuments dry up and the ovule is converted into a seed. The process of fertilization causes changes in the flower. All parts of the flower except the ovary wither away. The ovary containing the seeds begins to enlarge and forms the fruit.